Aloha. Today I will focus on transcription, or RNA metabolism, as chapter 26 is titled. The process of transcription is, in a way, the beginning of gene expression. Once the cell decides, or once the cell receives the signal asking for a certain gene expression, what the cell does is makes a copy of a certain part of the genetic material in the nucleus and exports that copy in the form of mRNA to the cytosol, where now the mRNA will be used as a template to synthesize a certain protein. Then that protein gets processed if needed, and it becomes functional to provide whatever function it is supposed to provide. So in a way, transcription is the first stage in the gene expression process that has a goal to produce a desired protein. Now, whenever we talk about RNA synthesis, I usually start with these questions. The first of those questions is how is RNA similar and different from DNA? And by now, I'm sure you already know that RNA, the main difference between RNA and DNA is actually in the name of the nucleic acid. One is deoxyribo, while RNA is ribonucleic acid. And that tells us that all nucleotides in the RNA molecules contain a ribose that has a hydroxyl group bound to the second prime carbon of the pentose. In addition to that, while DNA nucleotides do not have that hydroxyl group bound to the second prime carbon. In addition to that, RNA contains uracil bases, while DNA contains thymine bases. DNA is double-stranded, while RNA is usually single-stranded, but that single strand reaches a unique secondary structure. Functionally, DNA is a storage of genetic information. RNA can also play that role, for example, mRNA. It carries that genetic information in a way, it stores it in its structure, but it also has a lot more, RNA molecules are a lot more involved in other functions, such as catalysis, transfer of genetic information, even functioning as enzymes. Well, I already said catalysis, but I mean in a slightly different way, and I will explain this a little bit later. So how many kinds of RNAs are there, and what is their function? Well, there are actually various kinds. Each one of them has its own name based on the function that they provide. For example, the most common one perhaps that you know is the mRNA, or the messenger RNA, and the function is to copy. Well, it is a copy of the genetic information that's going to be expressed, and transferring that genetic information from the nucleus to the cytosol. tRNAs, or the transfer RNAs, are the RNAs that provide the matching between the amino acids to their corresponding codons in the mRNA. rRNAs, these are the ribosomal RNAs. They are part of the ribosomes. They actually comprise the active site in the ribosomes. Then we have ribozymes, which are ribosomal enzymes. They don't have proteins. The enzyme is just a ribosome. An RNA. We have small nuclear RNAs, or snRNAs. We will see them in action when we talk about splicing. We also have miRNA. These are fairly recently discovered small RNAs. Mi stands for microRNAs, and they have regulatory function in the cell. And we'll talk about them a little bit later. So how is replication similar and different from transcription? Well, you might say, well, we haven't really talked about transcription yet. How should we know what is similar and what is different between these two processes? And in that regard, you're right. But I know that if you have done your preparation, you already have a certain level of understanding of both processes. And perhaps you can already answer that question. But if you cannot answer this question right now, ask yourselves at the end of this presentation, how are replication and transcription similar and different? At this point, though, I will just tell you that in terms of similarities, they both have a starting site. And they both have, for example, for replication, it's original replication, the starting site for transcription is promoter. And they all have stages, initiation, elongation, termination, that are controlled by different factors. The reaction that is involved in the synthesis or polymerization of the nucleotides is the same. The principle is the same. In both cases, we use polymerases, in one DNA, the other one RNA polymerases, and both of these polymerases are DNA dependent, so they need to have a template. The difference is the replication is a process that copies the entire genome. You cannot have partial replication, while transcription is usually partial. Only the part that needs to be expressed, the part of the genome that needs to be expressed is copied. The rest of it stays basically unaffected. Another thing that is different between replication and transcription is replication uses both strands of the DNA and uses them both as templates and synthesizes new nucleic acid or new DNA, while transcription uses only one of the strands that is called the template strand. And something else that is characteristic for transcription, and that is the fact that transcription does not have, or RNA polymerase does not require a primer in order to function, while DNA polymerase does require a primer. An interesting fact that I don't know whether you're aware is that most of the genome is transcribed. All mRNAs are known as transcriptome, but less than a third of the genome is transcribed for protein synthesis. So what is the purpose of the rest of the genetic information? It's a good question that you ask yourself too. Well, approximately 20 plus years ago, that extra DNA that was not coding for proteins was often referred to as the junk DNA. In other words, DNA that has no purpose. DNA can be just excised and thrown away. Basically, we just have it, but it doesn't really play any role. Or if it does, it's just a minor role. However, with time passing and us understanding more and more how the genome functions, we began to understand that this is not really a junk DNA. Much of that so-called junk DNA or excess DNA contains regulatory sequences or parts that are that control the expression of other genes. And of course, there is a large portion of it that we still don't quite understand what is its purpose, but I highly doubt that it's simply junk DNA. All right, so let's now start with the parts required for the transcription process. I will begin with RNA polymerase. It is an enzyme that binds double-stranded DNA, but once it is bound, it uses only one of its strands as a template, and I will expand on that one a little bit later. It does require a template, it does require nucleotides, and it does require ions such as magnesium and zinc. It does not need a primer, but it does start at a specific site known as the promoter site. And that site has a binding element that recruits the RNA polymerase to bind to it. Once the RNA polymerase is bound, it causes a partial unwinding of the double-stranded DNA. It's about 17 nucleotides. And this partial unwinding of the DNA is known as the transcription window or transcription bubble, whatever you want to call it. And like I said, one of the strands is used, and the RNA polymerase starts synthesizing based on that template strand, the new RNA or mRNA molecule. So approximately 8 nucleotides are basically span this hybrid between DNA and RNA. You can see this double-stranded area where DNA and RNA are actually bound together. And after that, RNA comes out, and when it's finished, it's just basically gets transported out and used for whatever purposes it needs to be used. So again, no primer. RNA polymerase starts immediately from the place where transcription begins and starts synthesizing the new mRNA, and approximately 8 bases exist as a double-stranded hybrid between DNA and RNA. The synthesis of RNA, similarly to the synthesis of DNA, is always in the 5' to 3' direction. The new nucleotides will be added to the 3' end of the newly synthesized RNA. The reaction, the chemistry of the reaction, is exactly the same as with the DNA polymerase. And the hydroxyl group bound to the 3' carbon attacks the alpha phosphate. Pyrophosphate is released and then immediately gets degraded to inorganic phosphate, and phosphodiester bond is formed, and that's how the polymer RNA grows. So in that regard, no differences with DNA. Alright, so obviously RNA polymerase will synthesize a transcript or mRNA. So it is important to know what the sequence of the product would look like as a result of transcription. So if I give you a sequence such as this one, what would be the sequence of the transcript of this DNA? Now, many of you would probably wonder, well, how can we tell? I mean, this is just a stretch of DNA. We don't know the orientation. We don't know whether this is the sense strand, whether this is the template strand. How can we tell? Well, whenever a sequence of DNA is given to you and nothing is specified about it in terms of direction or any other information, you are safe to assume that the sequence given to you is in 5' to 3' orientation. And as a result, that means that this is the coding strand. which automatically tells you that the antisense strand that is not shown here is the template strand. And the antisense strand, the 5' of antisense strand will be here, and the 3' will be on the left. In other words, the strand will be read similarly to DNA polymerase in the direction of 3' to 5', while the newly synthesized RNA will be synthesized from 5' to 3'. So if you can imagine, the template for the transcript synthesis will be the antisense strand. And the product, the complementary product to the antisense strand, would look exactly like the coding strand given here, with one difference, well, two differences. All nucleotides will be ribonucleotides, and all T bases will be uracil bases. So the answer to this question is the sequence of the transcript would look exactly like the coding strand, but the nucleotides will be ribonucleotides and all T's will be U's, or uracils. Alright, each double-stranded DNA has a template and a coding strand. Which one is given above? Well, I already answered this question, the coding strand. Alright, something interesting for you to realize is that not all DNA in the cell contains genes that are expressed in the same direction. In other words, that the antisense strand is always the template strand for all genes, if we can represent the genomic information in this way. Each strand can be expressed in both directions. In other words, if the beginning of a gene is here and the end of the gene is here, represented by each one of these arrows, that means that the promoter for this gene will be somewhere here and the termination signal will be somewhere here. This will be the coding strand, or the 5' to 3' strand. That means that the antisense strand will be the template strand. However, if the beginning of the gene is here and the termination signal is here, that means that the gene will be expressed in the opposite direction. So for such a gene, this strand that is shown here as the bottom strand would be the coding strand, while the top strand on this image will be the template strand. So in order for you to know what is the transcript, the sequence of the transcript, you need to know the direction of the expression. So if you're given a sequence like that, that means that the direction is this way, 5' here, 3' here, because always the DNA is given in 5' to 3' unless otherwise specified. Okay, so I want you to be aware of this, that the genetic material is not just always in one direction. Expression can happen in both directions. That's actually quite ingenious because we have a double use of DNA. You can have twice as many genes from one stretch of DNA if you can express it in both directions compared to if you just can express it in one direction. All right. The RNA polymerase, the core subunit of RNA polymerase, contains 2 alpha subunits, 1 beta, 1 beta prime, and 1 omega subunit. In addition to that, the core subunit binds to a sigma subunit. Now, this subunit is not permanently attached to the core subunit of RNA polymerase. It actually attaches transiently. It attaches and then it gets removed, and then another one can come in and be removed, and a third kind can come in and be removed. They're all sigmas, just slightly different. The rate of synthesis is 50 to 90 nucleotides per second, and the error rate is 1 in 10 to 1 in 100,000 additions. Now, do you recall what was the rate of the error rate in DNA replication? it was 1 in 1 to 10 billion bases. That is quite high, or the errors or that process is far more strict compared to transcription. And we already know that RNA polymerase has the ability to to repair wrongfully added nucleotides, but it is not yet clear whether this is the classic 3' to 5' proofreading activity. Nonetheless, though, wrongfully added nucleotides can be repaired. But again, we don't know whether it's 3' to 5' proofreading or something else. Now you may ask yourselves, how come that the errors in transcripts are far more common, and how will that affect the function of the body or the cell? Wouldn't that be still damaging to the cell? And if not, why is it less significant then for the body? Well, if you think about it, if a newly synthesized DNA has errors, every single copy from that gene, every single transcript from that gene, if it's in the gene area, will contain that error, every single one of them. But in the case of the transcripts, if a single mRNA copy has an error, I mean, it might be bad for the resulting protein, but the cell probably will be able to overcome that issue. Why? Because let's suppose that you make 1,000 copies of mRNA. If one of them, because this is 1 in 10,000, I guess, if 1 or 2 mistakes are present, well, the rest of the 998 don't have that mistake, and they will generate perfectly fine proteins. So the overall effect is masked by the correct copies of mRNA that will be generated. In addition to that, if a protein is synthesized and that protein doesn't fold properly, or for some reason there's something wrong with it, and it doesn't get localized to the right location, or like I said, folding, it might be recycled and degraded, and then after a while, that mRNA copy would have spanned his life, basically, and it would have been degraded, and the effect on the body will disappear. These transcripts have a half-life of a few minutes to maybe about a day at the most. Usually it's about an hour to 2-3 hours. So you see, even if you make a bad copy, it won't last very long. It will be eventually degraded. But if you make a bad copy of the genetic material, that will stay as long as the cell is alive, and anything produced out of that genome will contain that copy, that mistake. Now, transcription begins, like I said, at a promoter site. So having in mind that thousands of genes need to be transcribed within a single cell, what properties should a promoter site has? What would qualify a stretch of DNA as a promoter site? I mean, can you imagine? You will use pretty much the same machinery to express thousands of genes. How can the body or the cell distinguish which genes are supposed to be expressed now, which genes later? Which gene should be expressed in a neuron, liver cell, muscle cell, skin cell, hair follicle? Which gene should be expressed? How can the body determine what's to be expressed and what's not to be expressed? I mean, that's I was going to say that's the $1 million question, but that's more like that's a $1 trillion question, because once we understand that, either we're going to make great advances in medicine and therapy, or we will unlock a great evil in the world, because the ability to control expression is a tremendous power, and as you know, men are so prone to power, to want more power, that once they understand such powerful processes, inevitably somebody's going to make a bad use of them. But this is one of the fundamental questions of gene regulation. How can the body or the cell control that expression, the expression of its own genes? Because the genetic material is exactly the same in every single cell, while the cells have very different function. Each cell has a very different function. Each cell type, I should say. But let's get back to the promoter side. So, what property should a promoter site has, and what would qualify a stretch of DNA as a promoter site? So, again, promoter has to recruit the transcription machinery. In other words, it has to have sequences that are similar between all promoters. And these sequences are known as consensus sequences. They are not identical, but they are very similar. These are binding sites for the proteins responsible for transcription. So some binding sites will be the same or very similar for all proteins, because that is where, because they're the same proteins for all transcriptions, such as RNA polymerase. So the RNA polymerase site should be fairly similar for all promoters. Some will be different though, because these differences will add the diversity in the promoters, so that it will allow for differential expression. Such as in bacteria, in the case of bacteria, the binding sites for the sigma. Or in the case of eukaryotic cells, as we're going to see next time, the binding sites of the transcription factors. Not all promoters will have the same transcription factors binding sites or the same sigma subunit binding sites. Also, the variation within the sequence, within the site, also will determine that variability in the binding, in the strength of the binding. Some sites will have higher affinity for a sigma or for the protein binding. Others will have weaker. If the site has higher affinity, that means that site will be bound and transcription activated at a lower concentration for the sigma subunit of the transcription factor. If the binding site has a sequence that has a weaker affinity for the sigma or the transcription factors, that means that a higher concentration of these proteins will be required in order for this site to be expressed. I mean, the sky is the limit in a way in terms of selectivity, and it has to be if we're so profiled. If we have cells that do everything pretty much in the body, such as the liver cell, and then you have specialized cells such as neurons or muscle cells that have a very strict primary function. There has to be some form of selectivity and diversity within the promoters, so that the cell is allowed to select what to express and what not to express. So, in terms of bacteria, it's been determined that the bacterial promoters span the area from the plus 30 to the negative 70 nucleotides. Plus 30, if there is a RNA start site, that promoter will span 30 nucleotides downstreams and about 70 nucleotides upstream. And this diagram shows an example of promoters from different bacteria, and you can see certain consensus sequences within the -10 area, the -35, the UP element, that there is certain consistency between the sites. Someone would say, "Wait, they look quite different." I mean, you're right. They can be very different. I mean, look at this one. It's TA. There's absolutely no G in this one. But this one has 2 G's. This one has 1. This one has 1 C. And the T's and A's are not always in the same sequence. So there is variation, but there is a consistency or higher possibility to find these sequences in this area compared to any other. That's why it's a consensus sequence. Okay, well, I'm also saying here that the bacterial analysis of promoters has determined that the RNA polymerase binds usually in the -70 area sequence or segment, and that's the binding site where RNA polymerase usually binds. All right, how do we determine where the promoter is? Because there is no specific location for the promoter. You don't, I mean, for bacteria it was established to be +30 to -70, but in higher organisms, that's not the case. It's very difficult sometimes to find out where the promoter is. One technique that is used to determine where the promoter is or where RNA polymerase binds in order to express a gene is known as footprinting. This is a fairly classic methodology. I'm not sure whether it's still highly in use. Perhaps it is because it's so simple. But this is how it works. We have a sample of DNA and we separate it into 2. One is by itself. They're all labeled so that these red dots basically means that we've labeled them to a radioactive nucleotide so that we can trace them and detect them actually. One sample is just the DNA. To the other sample, we add RNA polymerase. And of course, anything else that is necessary for RNA polymerase to interact with its promoter site. So RNA polymerase will be incubated with this DNA and eventually will bind its binding sites. Then we add endonucleases to these samples, and either usually it's 1 or 2, but these endonucleases will start cutting everywhere they can bind within the DNA, everywhere there is a site that they recognize. And then each sample is resolved via DNA electrophoresis. This is a sample or the pattern of the digestion of the DNA without RNA polymerase. This represents the patterns shown in the presence of RNA. And we can see there that there is a gap in the distribution of the bands. Now, why is that? I mean, you can imagine here with the arrows is shown where the endonuclease will digest. If, for example, endonuclease has to digest here, or perhaps within the binding site, that endonuclease will not be able to cut anywhere in very close proximity to the RNA polymerase. Even if it's outside of the binding site, sometimes the proteins are so bulky that they don't allow anything else to associate with these sites. So there will be certain digestion events will be prevented from happening because of the binding of the RNA polymerase. Here is an actual experiment where RNA polymerase has been incubated with a lock operon. This is the control. It's just basically sizes. This DNA band sizes. This is without the RNA polymerase, and this is with the RNA polymerase. Notice that certain fragments disappeared, some remain, and some fragments appeared, because if you change the binding or the cutting pattern, then some fragments will change their size and new fragments could appear. And knowing what those predictable fragments are and where the cutting sites are, then you can determine where RNA is bound, because those are the sites that were not cut. Like I said, this is a classic method. More modern methods are the so-called primer bashing. I'm not sure how official that name is, but at least that's what we call it. Not primer, but promoter bashing. For the interest of time, I'm going to go a little quicker through these figures. Actually, the time really is not a factor here because everything is online, so I can allow myself a little extra time. But anyway, let me explain how this works. We have expression constructs, and now you know what expression constructs are, and these constructs express luciferase. Luciferase has the ability, when excited with a substrate, to produce light. That light can then be detected by a special instrument, and based on the amount of light, we can judge how much luciferase have been expressed. Alright, these are special reporter constructs. They do not have, and luciferase does not have, a promoter in front of it. So if you just put the construct the way it is, the way you purchased it, nothing will happen. And these, like I said, these constructs or vectors are designed so that the investigator can insert segments of DNA that they suspect to have a priming capabilities, to contain a primer. Promoter. I apologize, I keep confusing primer with a promoter. If you want to study if certain segment of DNA has a promoter sequence, then you insert it right in front of the luciferase gene and then measure the amount of luciferase produced. So what do we see here? We see that if we insert this 1894 base pair segment in front of luciferase, we get a certain signal. If we remove the top 170 bases, then we get about the same. We continue to basically cut off small pieces from that DNA, from that inserted segment, and we seems that the signal is pretty good. It seems like actually it's increasing, which actually doesn't make any sense. Why lowering or decreasing the amount of nucleotides will increase the signal? That's actually could be quite the reason for that could be artificial, because constructs that are very large, such as this one, or even this one, the larger the construct, the more difficult it is for that construct to be transformed or transfected. The smaller the constructs are, the easier it is. And so as a result, you might be simply getting more DNA into the cells, and so you're basically generating more product. On the other hand, there could be certain inhibitory sequences in this area, or perhaps the fact that you have more does not allow, that you have more allows for the DNA somehow to interact with itself and that to inhibit the expression process. Whatever it is, this is not abnormal. Seeing an increase by decreasing the size of the inserted DNA, this is not what worries us. What worries us, at some point the expression, well, I shouldn't say worries us, but what is the sign, or what is a sign that the sequence contains promoter sequences or functions, is when that expression starts decreasing. And so at some point we've reached now 387 bases from the original. And when we cut about 60 of those bases, suddenly we get about 40% decrease in the expression of luciferase. If we cut another 100 of that, we get half of that previous and 60% decrease from the 8387. And if we cut another 70 of that, it continues to decrease until it completely disappears when we are left with about 78 nucleotides. So, if you have to estimate where is the primer in this sequence, your best estimate will be between -78 and -387. That's where the primer is. About 300 base pairs between -78 and -387. Here's another example. I'm not going to go into much detail here, but you can see the same things, just a stretch of DNA with possible promoter capabilities is cloned right in front of luciferase, and this is the corresponding expression. Again, do you see this increased expression? I bet this is simply an artifact from the fact that the segment of the DNA decreases. In other words, transfection is it is increased with the decrease of the size. But at some point you start seeing a decrease in the function or the amount of luciferase right here after 696, and then from 456 to 150. But then once you remove 145 somewhere there, nucleotides, you lose everything. So where is the promoter site here? Between base 9 and 696. So it's about 600-700 nucleotides. And when we sequence now this segment of DNA, and we do search for consensus sequences, now we've identified, this is eukaryotic cell, we've identified binding sites for transcription factors, NF-kappa B, SRF, CMIB, ELK1, and again, NF-kappa B, you see 1, 2, 3 NF-kappa B sites. We see STAT, ELK1, 1 and 2 ELK1s, 3 actually ELK1 sites. These are all transcription factor binding sites. And it doesn't mean that every single gene will have this transcription pattern, a transcription factor binding pattern. Some will not have the STAT. Some will have different factors. Some will not have NF-kappa B. These do. Some will have 1 or 2 or 3 or more. And the binding of NF-kappa B to these sites is not the same. It could be stronger to this one, weaker to that one. You can see the complexity of gene regulation. It is enormous. Alright, so that's how we determine the priming sites. Now let's get back to the stages of RNA synthesis or transcription. The first, the transcription process, or the beginning of RNA synthesis, starts with the assembly of the transcription complex. And there's not much to assemble there. You will see later on with eukaryotes that it's a lot more complex than in bacteria. But basically what happens is this process is driven by the sigma subunit that associates with RNA polymerase and brings it to the promoter site. Once bound, the transcription window is formed by opening this 17 base pair window. It basically splits this segment of about 70 base pairs of the double-stranded DNA. And at this point now, the initiation begins. And what happens is the RNA polymerase starts synthesizing RNA and forms that 8 nucleotide long hybrid. The sigma subunit then is no longer needed because it did its job. It basically brought in the RNA polymerase to the gene that needs to be transcribed, and then RNA polymerase continues to synthesize the new RNA molecule, and we're transitioning to the elongation process. The elongation process is supported by the NUSA protein that binds, basically exchanges for the sigma subunit, and the binding of this protein basically supports the association of the RNA polymerase with its with the template, and allow more stable synthesis of RNA, until of course we reach the termination signal. When that happens, the NUS falls off, the mRNA is released, and then the RNA polymerase is now free to associate with another sigma subunit and start another round of RNA synthesis. Now, how is this process regulated? I mean, we've talked about the role of the sigma subunit, and what did we say? That the sigma subunit determines specificity. In other words, the sigma subunit determines where RNA polymerase will bind. That's in bacteria. We'll see in humans, but well, I can tell you, it's the transcription factors also that play a great role in the selectivity and determining which gene will be expressed. But how are the sigma subunits regulated? How is the regulator regulated? Well, different sigma subunits determine what genes will be expressed, like we've already said. Here we see a table how the different sigmas are involved in the expression of these group of genes. Now, the sigmas are usually regulated by various mechanisms. If we need sigma subunits to be expressed, then they will be expressed. So they can be controlled by expression. If we don't need them anymore, they will be degraded. So degradation. If we need them but not right now, we can turn them off, perhaps by post-translational modification or by sequestration. You put them in a certain area that they will not be available. Basically take them out of the nucleus so they're not available to perform transcription. And that can happen either with the help of other binding partners like other proteins, or perhaps some other mechanisms that just simply sequester the subunit away from the transcription site or from the transcription process where it occurs. The transcription is also regulated by other proteins that bind activator and enhancer and operator sequences. These are activators and repressor proteins that either stimulate transcription or suppress transcription. All right, and the last slide for this presentation is the termination of how appropriate the termination stage of RNA transcription. So in bacteria, there are two main mechanisms. One is the Rho independent and one is the Rho dependent. The Rho dependent is based on certain sequences within the gene. Towards the end of the transcribed sequence, the RNA basically reaches an area which is rich on adenine nucleotides. And the RNA polymerase, before that, prior to that A-reach sequence, there is an area that has the ability to form a hair loop. In other words, this is a palindromic sequence. And when that hair loop forms and the RNA polymerase reaches this A-reach sequence, it pauses. And then, with the help of this hair loop and the torsion that the hair loop causes, eventually this the hybrid, the DNA-RNA hybrid, will be broken, or the two sequences will be separated, and the RNA, newly synthesized mRNA, will fall off. Once that happens, the RNA polymerase also falls off, and we have terminated, or the transcription has basically been terminated. Now, the row-dependent mechanism is slightly different. It involves a C and A rich sequence, cytosine and adenine rich sequence. That sequence is known as the RUT sequence, or RUT sequence. It's not just an A rich sequence, it's C and A. So when RNA polymerase reaches that sequence, it pauses. And then the transcript, part of the transcript that is somewhat close to the CNA rich sequence has a binding site for a protein known as the Rho protein. The Rho protein then will bind the transcript, and it will start migrating towards the paused replication site. And when it reaches it, it will act as a helicase, splitting the DNA-RNA hybrid and releasing the RNA from the transcription site. And with that, termination is achieved. All right, so that is all that I wanted to share with you today. If you have any questions, come and see me during my office hours or email me. Thank you very much.